Now we're going to talk about our three groups of chordate animals, three primary groups. They are the Urochordata, the Cephalochordata, and then we'll call them the Craniata. Some books would refer to it as the Vertebrata instead of Craniata, but we'll get to that in a minute. The first group, the Urochordata, also known, or the most common form of this is what we call the tunicate. In the adult form, the tunicate is a sessile organism, kind of like a sponge that sits on the bottom of the ocean floor and filters food. Tunicate does a similar sort of thing. It has a different body structure completely than a sponge, but it has a similar kind of life on the ocean floor. The word tunic in tunicate refers to an outer covering that's on the tunicate structure. So anyway, Urochordata is one group, and then we'll have some examples of that. I've got a bottle. Here's a bottle full of tunicates. These are about the size of a grape and look like a very like grapes have been sitting in water for years or something like that. But anyway, that's tunicates. In the adult form, the tunicate does not have a notochord or a dorsal hollow nerve cord. It has a brain kind of structure, but it doesn't have that elongated feature that we see in the larval form of the urochordate. So it undergoes a kind of a transformation like a tadpole stage at the larval stage that has the notochord and nerve cord, and then when it converts over to the adult stage, that sessile organism, it loses that. So anyway, that's the Urochordata. Cephalochordata. The example, we usually refer to this as Amphioxus. It also goes by the name lancelet. But anyway, Cephalochordata, the Amphioxus. The Amphioxus, here I have a bottle of Amphioxus, a whole bunch of them in one little bottle. They're just a few inches long, 2 or 3 inches long typically. They are compressed laterally. That means that they have kind of a keel along the top of the body, and also on the lower part they're narrower. We'll study them in great detail in the lab, so you'll have to know the entire anatomy of a Amphioxus. The way that they live, they usually are in substrates, soft substrates on the ocean floor, where they will have their head slightly above the substrate and their body kind of buried down in the sand or substrate below. And they're filter feeders. What comes by, they do have some mechanism for kind of moving food into their mouth. But anyway, there's Cephalochordata, the Amphioxus. Next group, the Craniates, Craniata. As I mentioned before, sometimes we refer to these as the vertebrates. And used to be, and is sometimes still referred to as the subphylum Vertebrata. These are all categorized as subphyla of of chordates. Chordate subphyla, Urochordata, Cephalochordata, and Craniata. The hagfish does not have vertebra. So that's the reason why we don't include everything under Vertebrata. We changed it to Craniata because the hagfish does have a bit of a head skeleton, has cartilages, structures in the head, and the region of the brain So anyway, like a craniata. So the craniata includes these groups: the hagfish and the lamprey group. These are both eel-like in their shape. They're both aquatic, obviously. They're sometimes called the cyclostomes, which means circle mouth, or the agnathans. That means they do not have a jaw. So these are jawless fish, the hagfish and the lamprey. Hagfish is almost exclusively marine, and they are mostly scavengers on the ocean floor. Lamprey, some of you know the lamprey as a parasitic kind of a fish, and most of them enter into freshwater. Some are only in freshwater. Some kind of go between marine and freshwater environments. So hagfish and lamprey, these are agnathans. The next group in our list would be the chondrichthyan fish. Those are the sharks, the rays, stingrays, skates, if you know what those are, and another group we call the ratfish. So the chondrichthyan fish. Next we have two groups of what we consider bony fish. Bony fish, those are the ray-finned fishes. And the lobe-finned fishes. Ray-finned fishes and lobe-finned fishes, two groups we consider bony fish. So all of these are fish groups. They all have gills that allow water to move from the inside of the pharynx to the outside of the body. Following this group here, the next one in our list would be the beginning of tetrapods, the amphibians. Beyond the amphibians, we have the reptiles. Sometimes included with the reptiles are birds. So birds, sometimes considered a separate group, sometimes thrown in with the reptiles, and then finally the mammals. So these are the examples of craniates, where all of these have vertebrae except for the hagfish. The hagfish has the cranial skeleton. They all have cranial skeletons as well. Hagfish, lamprey, chondrichthyan fish, ray-finned fish and lobe-finned fishes, amphibians, reptiles, birds, and mammals. Those are your primary groups of chordate organisms. The three subphyla are Urochordata, Cephalochordata, Craniata. Next, we want to talk about phylogeny, ontogeny, and von Baer's law. These three topics kind of work together and are important to our understanding of comparative anatomy, an important biological topic. Phylogeny is a term that refers to the family tree of taxa. We've all seen the diagrams that show the relationships between different animal groups. We call these phylogenetic charts or graphs. Depictions of how things are related. Those things that are supposedly closely related, like two species of fox, would be close on the phylogenetic chart, while an elephant would be, their connection would be further down on that tree, that family tree drawing. Ontogeny is a term we use that refers to developmental process. This is also the embryology or ontogeny of an organism. So understanding how from the single cell tissues start to form and individual organs eventually start to develop, that's the developmental process. Von Baer's law links these two concepts, this idea of relatedness in a family tree kind of arrangement to the developmental process in here. So Von Baer's law, as I have it written here, anatomical features which are common to all members of a major phylogenetic group. Those arise early in embryonic development. So the early ontogeny, the early development of an organism, begins to show features or characteristics that are going to be shared by almost all members of a major phylogenetic group. While those features that distinguish subgroups of the taxa arise later. So those things that make dogs different from cats, those things that are separate those subgroups out more, they arise later on in the embryonic development, not until they're fairly far along do those more unique features start to be expressed in those groups. So, phylogeny and ontogeny relationship. They used to use a kind of phrase that says phylogeny recapitulates ontogeny, which means that the way that the family tree structure is developed kind of replays the process of development. So those features which are shared in all of the different groups, those come along early in the development, and they are lower in the family tree, shared among all members of the family tree. Those things that are more unique to subgroups, they would be developing later on in development and would be separating groups higher up in that family tree arrangement. Next, we'll be getting back to that phylogeny concept here in a minute, but kind of important, necessary parts of understanding how that works. We need to talk about conserved traits and derived traits. Conserved traits and derived traits. I have a definition here for conserved traits. First, traits that are assumed to be passed down from one ancestral line and shared by all or most subgroups of a taxon or a group. So their forebearers had it in the ancestral line, and all of the groups that follow that would have it. Next, those things that are derived traits are unique traits assumed to be of recent evolutionary development within a lineage and which differentiate one subgroup from others within a taxon. So those are the things that are new kinds of traits that show up in a phylogenetic illustration. So those are two important concepts we have to know to understand how to make a phylogenetic arrangement or how to interpret phylogenetic arrangements that are critical to our structure of understanding those relationships and the grouping of organisms overall. Okay, I mentioned that conserved and derived traits are vital to the construction of what we think of as a phylogenetic chart. Now, it's important to remember that this kind of grouping of organisms occurred even before we had any evolutionary theory. So people would understand things that had certain features like claws and fangs and so forth. They could kind of group together in one group of organisms, and they also had fur. And also cattle and other ungulates and such had fur. So they were also in that same group of organisms we now call mammals. So anyway, this kind of grouping of organisms according to shared or derived traits is nothing new. Let's see, on my board here I have a phylogenetic chart that groups different organisms together in a family tree kind of pathway. First of all, down here way at the base, everything that's present in these examples have venom, these are venomous snakes, with long hinged fangs. Venomous snakes with long hinged fangs. So whether it's considered a viper, rattlesnake, or a pit viper, they're all in this group. We can call it the group of vipers, the venomous snakes with long hinged fangs. Now we have a subgroup within the vipers coming out this direction, and everything on this path right here has a heat sensing pit behind the nostril. So between the nostril and the eye in there, there's that heat sensing pit. So we call everything on this lineage right here the pit vipers. Pit vipers. And within the pit vipers we have a subgroup all these have a tail with a rattle. So this is a type of phylogenetic chart. Vipers are snakes, we know that. They have things that they share with other snakes. Conserved traits among snakes, their elongated body, their lack of limbs, a whole suite of characters make them snakes. We know snakes when we see them, usually. So anyway, this is the one trait that sets off this group from all the other snakes. So this would be a derived trait if your frame of reference is snakes, venomous snakes with long hinged fangs. To make up the viper group, there are other kinds of venomous snakes that do not have long hinged fangs, things like cobras and coral snakes and mambas. They have their venom, but their fangs, their teeth that inject the venom are not the hinged type that swing out when they open their mouth. So anyway, that's a derived trait among the snakes, but it would be considered a conserved trait with respect to everything we have on the board here, okay? So a trait can be either conserved or derived depending on your point of reference. If your point of reference is all snakes, then this long hinged fang arrangement here is a derived trait. If your frame of reference is just these vipers and rattlesnakes, pit vipers, this would be a conserved trait for everything that's listed here. Conserved trait there. So, in this chart, the next thing up, the heat sensing pit, is a derived trait among the vipers. It's a new thing. So this is a unique trait to these subgroups over here. So here is the pit viper lineage. On that lineage, we have those that have a tail with a rattle. That is a derived trait among the pit vipers. So it's a new trait there. But if our only frame of reference were rattlesnakes, for example, a conserved trait would be having a rattle on the tail. So your point of reference determines whether or not that trait that forms the Y, the basis for the split, is conserved or derived. Now we'll be looking at a lot of examples of phylogenetic charts showing similarities and differences in the lectures to come. So we got to have this kind of concept down to understand what we'll be doing as we move through all the different taxonomy and phylogeny for all of the different chordate and craniate groups. Now, a familiar topic to most of you, what we call the hierarchical system of taxonomy, kingdom, phylum, class, order, family, genus, species, the way that we group organisms according to their similarities and differences, as we just talked about with our conserved and derived traits. Our phylogenetic chart kind of comes back to us here in our naming pattern for our different groups. So we know there are kingdoms like plants and animals and fungi and so forth. These kingdoms kind of changed since I was in school, so we've added some, and especially among the bacteria. Things are divided up differently now than they were back then. Phylum is the next group up, next more specific group when you're trying to classify something. Then the class, order, family, genus, and species. As just an example, there's a special fish we'll be making reference to occasionally in the course called Polypterus. Poly, P-O-L-Y, refers to many or a number. P-T-E-R-Y refers to a wing or fin-like structure. So this particular fish, it's in the aquarium right close to us here. It has basically a body shape like this. It has its eye here, and then on the back it has a whole series of what looks like small fins all the way down toward the tail. Back there on the back, it has some fins up here in the front and some other fins back here in the back. So anyway, there's our Polypterus. We can now take this example, this unique example of a fish. It's considered one of the most primitive of the ray-finned fishes and old in the fossil record. It's obviously part of the animal kingdom, right? So we have animal kingdom. Phylum. We've just been through all that. We know that it's in the phylum Chordata. It's the chordate phylum. It's among the chordates. And in the Chordata, the class would be Craniata. No, that's a subphylum, excuse me. The class is the ray-finned fish group, which is Actinopterygii. The order is Polypteriformes. So the entire order is named after that particular fish. Family. Also named after Polypterus, Polypterides, genus Polypterus. And there are about 10 different species of this genus, and the one we have here is Indolicari. Indolicari, I guess. Indolicari, Polypterus indolicari. So that particular type of fish we can group with these names here. And I mentioned a minute ago when I was going through here, I made the mistake of putting in the Craniata as a group. That's actually a subphylum, which for any of these you might find occasion to include an intermediate group, and we'll talk more about that later. So there can be subphyla, there can be superclasses and subclasses, superorders and suborders, subfamilies, and so forth that people might use as taxa to help us group and understand the relationships of the different organisms, similarities and differences between the different taxa.